Hello, welcome everybody, and thank you all for joining us today for our virtual event. My name is Dr. Alan Mullen, Product Specialist for Microscopy Cameras at Andor, and I'm going to be your host for today's live presentation. In this presentation, we're going to be talking about the use of Actiphage to improve our understanding of mycobacterial diseases. So I'm delighted to welcome today's speaker, Dr. Kath Rees. Dr. Rees is currently Associate Professor in Microbiology in the School of Biosciences at Nottingham University at the Sutton Bonington campus. Originally, she studied biochemistry at Oxford, followed by a PhD in bacterial genetics at Leicester University, and has research focused now on applying molecular techniques to the study of applied microbiology, with the specific interest in the study of listeria and mycobacteria in food and agricultural systems. Recent research has resulted in the development of rapid phage-based tests for the detection of the important mycobacterial pathogens, including tuberculosis in humans and paratuberculosis and bovis in animals. And this has led to the establishment of PBD Biotech Limited, where she also has a role of Chief Scientific Officer. In 2019, the company was awarded the Royal Dairy Innovation Award for Research and Development in the field of dairy farming, and more recently won the British Veterinary Association 2021 Innovation Award. So just a little housekeeping before we get started. Everyone has joined the presentation in listen-only mode, so if you have any questions during the presentation, please type them into the question box in the control panel as we go along, and then questions will be answered at the end of the presentation. So now, without further ado, I'll hand over to Dr. Rees. Thank you.
Thank you for that introduction, Alan. Okay, so this afternoon I'm going to talk to you about using viruses to detect bacteria. Again, I guess this is slightly off-field for the rest of the sort of virology theme of the conference. So I'm not going to spend a lot of time talking about the viruses, but more how we've used those to try and understand these mycobacterial diseases, which are ridiculously difficult to work on. So as Alan said, I also have a secondary role to my academic post in a spin-out company called PBD Biotech, who sponsored a lot of this research. So just briefly then, introduce you to our viruses, the bacteriophage. They are viruses that specifically infect bacteria. On this first slide, panel here, you can see an agar plate where we have a lawn of bacteria all over the surfaces, and we can see the zones of clearing where the plaques are forming, where the viruses are killing the cells. So they were originally discovered and given this name bacteriophage from the fact that they could eat bacterial cells. So like all viruses, the first thing they have to do is bind to and recognize their host cell. And there's some fabulous helium-ion pictures that came from a Finnish group. And unlike eukaryotic viruses, to achieve this, our viruses usually have these tail structures, which encode enzymes that will help them break open this thick cell wall. Now, this thick cell wall is one of the reasons with mycobacteria we have so much difficulty working with them. Unlike a lot of sort of the standard eubacteria, the gram-positive, gram-negatives we're all used to, the mycobacteria have a thick layer of mycolic acid coating their surface, part of the surface structure, which means they are very, very robust and very difficult to break open by conventional methods. So we have all of these evolved structures and enzymes to puncture the cell wall, get the virus to insert its nucleic acid into the cell, start replicating, and then, like many of the bacterial viruses, it ends in cell destruction. So the phage make enzymes which are specifically evolved to break these cells open from the inside out. Now, what we discovered, and has caused a bit of a game changer for us, was that during this process, this particular virus does not degrade the host cell DNA. And so during this lysis event, the DNA from that host cell is preserved, and we can then use this as a DNA extraction method to recover DNA that we can use to detect using conventional PCR-based methods. So this is a very brief summary of the methodology we developed using these phage to develop our phage-based method, which we combine with PCR for identification. So we're looking at blood samples primarily, and what we know about mycobacteria is they replicate inside white blood cells. So the first stage is to purify the PBMCs. We can use Ficol, we can use other more modern techniques such as Hetasep or differential lysis. It doesn't matter, as long as we recover intact white cells and separate out those from the rest of the blood components, which may interfere with the assay. So those purified white cells, we then break open to release the mycobacteria, which are inside them, into a growth media that allows the mycobacteria to start metabolizing. We add the phage, let them do their work. Now, in the original format of the assay, which we developed approximately 10 years ago now, there was an intermediate stage where we got agar plates, as I showed you in that first slide, where plaques would form, and we could therefore enumerate the number of cells present in a sample based on the number of plaques detected. And then from those agar plaques, we extract DNA and do the PCR-based identification. Oops, all right, so our first test was developed for detection of bacterial infection, paratuberculosis infections in cattle, Johne's disease, which is an important economic disease for the farming community. And these typical tests that you had for the standard diagnostic for this was, one of them was culture, which would take up to 40 days. So to develop a test that took 48 hours was a massive improvement at this stage of the process. So to show, once we developed the assay that it would work, we went out to look for field samples. So went to a herd of cattle with a known Johne's disease problem. And we also sourced a high status control herd with no known history of disease. So in our infected herd, we were looking at cattle based on the milk ELISA results, which then it's another test that's standardly used for diagnosing Johne's disease. The only problem with this test is known to be quite insensitive and for the results to fluctuate. So the cattle were chosen to give us the best chance, really, that had 3 consecutive positive milk ELISAs, so consistently giving a positive result. So these are given a red status by the scoring system. So we took the blood samples for the MAP ELISA, also for a commercial direct PCR test using a kit produced by a company called Tetracore, and also for the blood phage PCR assay. And this was the result. We only had a few animals in the first trial. We had 9 from the Johne's herd, and we had a control set here of 5 animals. And as you can see, we had the milk ELISA status, okay, even though these had been chosen on the fact they were red, this result was starting to fluctuate. And when we did the blood ELISA to compare with the milk ELISA, again, we saw most of the animals were positive, but we were then starting to get this fluctuation. When we did the phage PCR, though, we got all positive results. Now, because we were using the assay where we could count the number of cells, we knew that in each of those 2 ml blood samples, there were less than 35 cells per sample. In the negative herd down here, we could see that all of the negative animals were negative by phage PCR, by blood ELISA, by milk ELISA. So we got good agreement across these tests. But when we looked at the Tetracore results, everything was negative. It didn't matter if it came from the positive herd, where we knew we had these low numbers of cells, or from the negative herd, everything was negative. But as you look at the test, as I said, these mycobacteria are very difficult to break open by standard DNA extraction procedures, and the quoted limit of detection for that kit was 100 cells. Well, as we've been counting less than that, we knew that the technical likelihood that these would be positive samples was low anyway. So that was just sort of the first snapshot where we went into and chose red, definitely red, and definitely negative animals. In a second follow-up study, what we did was to also have red cattle, but we also chose some of those cattle who were giving fluctuating results, so were either amber or green scores. So green meaning they'd never given a positive milk ELISA, and amber meaning that they had occasionally given a positive milk ELISA. Now, again, when we looked at these results and looked at the blood ELISA, in this red group here, you can see we're still not getting agreement between these two antibody-based tests, both looking for MAP, one looking using a milk ELISA, the other using a blood ELISA. When we did the phage PCR, though, again, encouragingly, all of our red animals were red. And interestingly, this time, one of the amber animal and one of the green animals also gave a positive test result. So we were getting to the point where we kept on seeing this discrepancy between antibody-based results and quite a lot of positives, even in negative animals, with the phage test results. Again, just as a comparator, we knew that the tetracore wasn't working, so on this occasion we tried culture. Everything was negative. Again, less than 35, in fact, actually less than 50 cells per mil in this set of animals. And because of the way that you carry out culture with mycobacteria, we have to incubate them for long periods of time. We have to carry out a chemical decontamination step, which reduces the viability of the mycobacteria as well. So even for culture, we have a quoted limit of detection using decontamination procedures of about 100 cells. So again, we wouldn't expect these to be positive. But this was now very encouraging results because we were getting nice, consistent results that made sense in the context of the disease. And they seem to be more sensitive than culture or the antibody-based tests. So let's just go back and look at the antibody and the immune responses and what's known in cattle about the immune response to this disease. And it follows quite an unusual pattern. And this is seen also for Mycobacterium bovis infections in cattle causing bovine TB. What we know is that at the onset of infection here, the first responses are going to be with the cellular immune response at all, with virtually no antibody response being detected at all. So antibody production is known to occur later on, and it often coincides with the point where the cellular immune response is starting to go into a decline phase. When the antibodies levels rise, they rise quite slowly, and it's often seen that it's just before clinical symptoms start to appear that you get a positive antibody ELISA result. And in the meantime, these cellular responses are still declining. So it's not overly surprising that you're getting disagreement between different types of immune response-based tests. In terms of the ELISAs, we know it's a slow increase, and you get this sort of late rise to the point where we normally get high levels of antibody that can be detected. However, there is also a feature known that this does not actually go up in a linear manner. It tends to fluctuate over time, often in response to the sort of environmental stresses that animals are exposed to. So stage of calving, changing of housing, moving between social groups, this sort of thing will cause the antibody levels to fluctuate. So in 2017, the Animal Health Research Group in Ames in the USA, in the USDA Animal Health Group, produced this lovely paper where they really set out to compare the way that different tests were performing. And there was two things that were important about this study. First of all, they recognised and really set out the fact that when you get paratuberculosis infections in cattle, there are two different forms of disease identified. And those are slow and persistent infections and fast progressing infections. And what they saw was when they looked at the efficacy of the different tests in these two different types of disease progression, they were different. So they've sort of divided up the disease into early, middle, and late, sort of as we progress along that graph of infection. And you can see in the slow and persistent infections, the ELISA tests were unreliable but detectable early on and then disappeared and then came back later. Fecal culture, so excreting the organism into feces, was always at a low level, below that limit of detection I talked about because of the need for decontamination. And even the cellular responses, which came up early, disappeared quite quickly and then were no longer useful. In comparison, if they looked at these fast progressing infections, so you could see that the ELISA stayed more useful for longer. Fecal culture became very strong in the late stages of infection. But interferon gamma were actually stable, but intermediate and unreliable all the way through infection. So this is why in sort of agriculture, they're looking for these new tests, because you have this fluctuating pattern. You have to take repeat tests to see if you get a positive result. And if you've got different forms of disease at different stages, you may get different types of results. So what about our phage test? How reproducible is that? Well, at the moment, we still haven't done the sort of longitudinal studies we really want to do on infected animals, but we have been looking at how, from sort of parallel samples, a test performs. And we can see here the analysis of a set of samples from different animals, so sample 1 and sample 2. And again, we're just counting the number of cells detected by counting the number of plaques. And we can see we've got good overall agreement between these parallel samples from the same animal with an R-squared value of 0.91. However, if you look in more detail at the data, down at the bottom end here, when we get these very low levels, then we will get a bit of stochastic variation in the result. And we're talking about 5 bacterial cells per mil of blood, at which point, if we take 2 mils out of an animal and hope that those 5 mils are evenly distributed in every 2 mil sample, it's very unlikely that we would get an even distribution. And so we're not worried about this sort of bouncing around here, but that is often what we would expect to see with these very low number of samples. We would expect to see some fluctuation in our results. So, like all companies who want to sell diagnostic tests, we don't really want to do agar plates and have things in the laboratory where we have to incubate plates. And so I had a student who was sponsored by the company to reformat the assay and to turn it into something that could be carried out in using sort of real-time analysis. So this is the new, what we call Actiphage test. Again, the first part is all the same. We're just preparing the PBMCs. And as I've said, it doesn't matter which method you use to prepare PBMCs, as long as we store those cells in conditions that do not inhibit the growth of the mycobacteria. So at the moment, we're dependent on fresh blood samples because we can't use stored samples where the mycobacteria have maybe been treated with chemicals to preserve the PBMCs. But that, if it affects the viability of the mycobacteria, our assay is not going to work because the phage is only going to replicate in these actively growing cells. So blood samples prepared. They're broken open. The white cells are broken open, and the cells are released into the media to get them metabolically active. We add the virus. We wait 3 hours. That's our DNA extraction done. Clean up the DNA on a column. Run the PCR. So the whole test can now be done in 5 to 8 hours. So we showed, we could do this now within a 1 working day. It's much more high throughput, formatable, as it were. And we will also predict it's going to improve sensitivity because rather than moving between our sample into an agar plate and then doing DNA extractions, we were then going to have everything contained in one sample. So even if we had those low number of cells, we would expect to reduce sample loss and improve sensitivity with those low cell numbers. So, small set of experimentally infected animals using this new method, using paratuberculosis, was carried out with the Morden Research Institute in the UK. These animals have been infected, and infection was confirmed post-mortem by taking tissue samples and culturing. And in fact, most of the animals, 15 in this positive group, were infected, but there was one where they couldn't get evidence through culture of infection. And when we look at the Actiphage results, we see a good correlation here. The negative animal was negative, and most of the others, 13 out of these 14, gave a positive phage result. Now, of course, this culture took up to 40 days, and we got this result in the same day. Again, we carried out both forms of tests. We did the Actiphage, but we used the enumeration method just for comparison. And again, we are only detecting low numbers of cells, with 15 cells per 2 mL of blood being the highest number we were detecting. But what was interesting on this occasion was that they were all blood ELISA negative. Now, if you go back to that model of that fast developing disease, which we see with the experimental infections, we would have expected the ELISA results to be positive. And this just reiterates the problem we have with these mycobacterial diseases, that the infection models, where you're using high dose, seems to go through a different disease development pattern than we see in naturally infected animals. So, conclusions just from this little part is that we knew that we could use our phage PCR methods to detect low levels of MAP in the blood. Obviously, it's going to be more reproducible when we've got higher levels, but the results were looking very specific with very few false positives. At low levels, we are going to get stochastic variation in the number of cells per sample, but we have a very low chance of getting, which will lower the chance of getting a positive result from 2 mL of blood. However, we also were seeing this pattern where the Actiphage could detect infection before an immune response was developing. So this was the paper where we published this new method, and we didn't just do Johne's disease. We also looked at bovine tuberculosis in this paper. And just to show you a quick snapshot of those results, we did a small study using cattle on an annual test program for bovine TB. And this meant that it was very unlikely we would have animals in an advanced stage of infection. They were all going to be caught within that first year of infection. The blood samples were obtained immediately after the first positive skin test result on a farm. So we were looking at new breakdowns, new infections, again, looking for early stage disease. And when we did this, 95% of the animals that gave a positive skin test for the bovine TB also had detectable levels of M. bovis in their blood. Now, that was far higher levels than expected, because we're still working on this model of bovine TB being a lung infection primarily, transmitted through inhalation of aerosols, the disease is being established in the lungs, and then being transmitted to, disseminated to the other parts of the body later in infection. Again, our negative animals from another farm were all negative, so we had good confidence that this result was specific. Okay, so we'd move from paratuberculosis to bovine tuberculosis in blood, which then moves us to the most recent studies we've been doing in collaboration with Leicester University, looking for TB in the blood of humans. So in the clinic, they signed up volunteers to have blood tests taken. And in this first proof of principle study, there were 18 pulmonary TB group And 75% of those were found to have detectable levels of mycobacterium tuberculosis, now human TB, in their blood. In the control group, which was 5 non-TB acute respiratory illness patients and also 28 volunteers, everything was negative. And then the most important group for us, most interesting group for us, were the TB contacts that they were screening. So people who'd been in contact with someone with a diagnosed case of pulmonary TB to see if there was any evidence of infection. Now, in the IGRA negative group, we found no positive results, but in the IGRA positive group, those that were giving a positive reaction to interferon gamma, there were 3 gave a positive phage result. Now, after 7 months, 2 of those 3 patients progressed to active disease. Now, but in the intermediate time, they'd been screened regularly, and there was no evidence of disease, and it's only after 7 months, and it was only 2 out of these groups here that actually gave disease, and no of the other contacts in this IGRA positive group progressed to active TB. So we're very excited about this. This may be an early signature for conversion from an exposure to the disease and control into a signature of active disease, and we're carrying out work to follow that up. Now, interestingly, and this goes back to the map, generally speaking, if we look at the number of phage we were detecting in the different groups, we can see that the positive samples gave an average number of cells detected of 43.6 cells per mil, whereas our control samples, very few mycobacteria detected, 0.25. However, occasionally we got some samples that had high levels of plaques, but were PCR negative. And it didn't matter if we used the Actiphage method or the plaque PCR, these were negative. Okay, so these didn't fit the pattern, and these were also negative by all the other clinical tests for TB, but had clinical symptoms of TB, and they were classified as what is known as sarcoid. So they are treated with the standard cocktail, and they normally recover. Now, I just remind you that our assay for TB using this is exactly the same, and all we're going to do is change the PCR. So in this case, what we did was to take the DNA from these samples and look for other types of mycobacteria. So we started looking for Mycobacteria avium using a signature sequence, which is IS3111, which is present in all of the avium group, including paratuberculosis. And we got a positive result. Now, this DNA, IS31, has polymorphisms, which you can detect using restriction enzyme analysis. And when we did this, the samples from the humans, the sarcoid patients, gave the pattern that is associated with paratuberculosis. So we change our one sample that gave plaques to positive for IS3110 and identified now as being a patient that was infected with paratuberculosis. Okay, so that's me done. I've run a little bit long. So just to finish, to summarize what I've said, we now know that these phage-based assays can be used to detect low levels of viable mycobacteria in blood samples. We know that the cells are growing inside the PBMCs, and it's assumed that these represent macrophage that are being disseminated from the primary site of infection, but we still haven't done the work to to prove that. Okay, so we still have to confirm which are the infected types of cells. If you're interested in this sort of dissemination of these tuberculosis diseases, there's been a nice review published just last year, and I've given you the details there. For bovine TB and human TB, these results have gained new insights into disease progression. For bovine TB, the fact that the bacteremia develops much earlier than expected, and in human TB, potentially this signature of this transition from exposure to active disease. And also we have some preliminary evidence that we can detect MAP in human patients in these patients are classified as sarcoid, and that patient did improve when we're treated with standard TB antibiotic regime, which will cross over and affect a lot of different types of mycobacteria. So that's me done. Just before I finish, I want to flag up a paper that has come out this year. Okay, it's 2020. In fact, I think that's 2021. I think that might be an error. But this is another group in the across worldwide, a collaboration where they've been looking at different methods to look for paratuberculosis in blood, primarily using culture. And they did find a lot of positive samples. Now, this is a little odd compared to the results where we've seen, because they actually found that in their Crohn's disease group, they had up to 60% positive samples for culture. And also the phage assay was detecting high numbers of paratuberculosis, not only in the patient groups, but also in these non-patient groups. Now, they use a slightly different method to us, and it's slightly strange because in our control groups, we didn't see any plaques at all in our healthy individuals. And so there is a difference going on here somewhere, which has yet to be explained. Right, I have really run over now, so I'm sorry about that. But I will put up my acknowledgement slides and finish there.
Okay, thank you very much, Dr. Rees. That's certainly very interesting, and another interesting angle to pull in the Crohn's disease side of things as well to this. We've got quite a range of different questions that have actually come in for you. So if we'll now go ahead and see if we can get through some of these. One of the questions is, you mentioned testing milk samples. Have you tested and compared both pre and post pasteurized milk using this technique?
Yes, we have indeed. We published a paper on this about 5 years ago now. When you look at pasteurized milk, in retail pasteurized milk, we could find low levels of MAP in about 10% of milk samples on retail milk samples. We have done some studies looking at pre and post pasteurization and did see a good drop in most cases. We did find some evidence that in some pasteurization plants, there may be evidence of biofilms accumulating and post-pasteurization recontamination of samples. But we haven't published that part of the study because it was only because it was just 1 or 2 and we didn't have a chance to go back and do follow-ups because it was just a small test study. But yes, certainly in retail pasteurized milk, we can use this method to detect round about 10% of retail pasteurized milk, very low levels, less than 20 cells per 50 mils. But once you're consuming a lot of milk, that leads to a significant level of exposure.
Another question then is, can your test distinguish between live and dead TB?
Yeah, absolutely. And this is the point I was trying to make with, we have to have those mycobacteria alive and get them into some media to get them metabolizing, otherwise the phage won't infect. Now, like many viruses, these viruses have a recognition system, whereas they detect the metabolic state of the cell as they bind to the surface. There is a change in cell phage interaction depending on the charge state of the membrane. So if the cells are not viable, the phage don't infect, they can't replicate, they can't break the cell open, so we don't get any positive results with dead cells.
Another one is, I think you have covered this a bit, but it was, can this method be used to test for mycobacteria and other types of samples?
Yes, we are working on it. Because the viruses are phage particles, which are protein shells, they are sensitive to inhibitors binding to them and also to inactivation by things like proteases. So there's a lot of sample prep work we're doing to work on different samples. But yes, we have done a range of different samples. Fluid is usually best.
And there's a couple of fairly more general questions. One of them is about what is the treatment for livestock currently? Is there treatment for livestock, or do herds or individual livestock have to be culled?
Yeah. So obviously for bovine TB, it's a notifiable disease and the animals are sent straight to slaughter. For paratuberculosis, the dairy industry is really working hard to encourage farmers to reduce levels of infection on farms. And so there are sort of screening programs coming in and farmers being encouraged to try and stop vertical transmission on farm and reduce numbers. It's incredibly difficult. And because it's just an economic disease rather than a genuinely confirmed zoonotic disease, there isn't the financial incentive for the farmers to cull other than pressure from the food producers to say we want levels to be lower. And we're hoping that these types of tests can help farmers start to identify clean animals and improve levels of infection on farms.
Another a related question at that then is there a vaccine possible for this? Or does this have issues to do with cross-reactivity with tuberculosis, for example?
The only vaccine exists that, there are some killed vaccines. The original vaccine was a live vaccine, attenuated strain that was never really brought into commercial use after about the 1950s when that kind of went out. And there is concerns that it may cross-react with the skin test. So for paratuberculosis, in other species apart from cattle, they are going down that route, and there are some non-viable subunit vaccines that can be used. But then if you're only tested as an ELISA test, everything is positive, so you can't screen for the disease, and that complicates the only disease control program. Again, what we're hoping is that our test will act as a DIVA test and differentiate between vaccinated and infected animals, because immunology doesn't play a part in our response. So we have done a little bit of work of this and shown that in sheep, we can actually pick up infected animals that have been vaccinated because the vaccine doesn't have a very high efficacy. So we're hoping that's another application of this test as well.
So if we'll just sum up really in this with the last question, you can really see that you've, with the impact of this to do with the lack of effective vaccines, treatment for livestock isn't really an option, and you've managed to get around the limitations of PCR and ELISA-based methods, allowing for that better detection of animals that may be pre-symptomatic or asymptomatic. So I think the last question then that we can take for now is, when is this test going to be commercially available? Is it available now or at some time in the near future?
The company is doing a little bit of small-scale testing, but we've just received funding to go through a validation process, which if you want to use this for bovine TB, it has to be fully validated. And as part of that, we're also going to do a full validation for the paratuberculosis in cattle. Now, it's a very small little spin-out company, and we can only do so much. So there is available some small-scale testing, but we're mostly working with partners who are providing us with additional data so we can get more information for people about when tests are useful and what it will tell you. Because without those longitudinal studies, which, we need the big BBSRC grants, we need the big funding, we need the time, we can't answer a lot of the questions about, like on that little diagram with when the tests are useful and what's happening at different, we haven't got that data yet. So a little bit of testing is available, but for the human stuff, only through the Leicester hospitals.
Okay, well, thank you everybody who's asked a question so far. And if anybody has any other questions, they can still submit those questions throughout the event, and anything that we haven't been able to answer now and any of those extra questions, we'll make sure that we get those through to Kath and she can can get back to you on those. So recording of this webinar will be available then in the Learning Centre as well on our website from next week. So please share that with any colleagues that you may have that might be interested in this topic. And thanks then to Dr. Cafferys for this interesting and informative presentation, and to everyone that has taken the time to enjoy us. So I hope you all continue to enjoy all of the other presentations on offer throughout the event. So thanks everybody again, and goodbye for now.
Thanks, bye.
Thank you, bye.